Students, today we are going to learn what are all the diseases that affects the wheat crop. All of us we know the wheat is one of the staple food crop and it serves or nourishes, take care of the food need in India, which is affected by several diseases of them. The rust, there are three different types of rust which are predominant on the wheat causing economic yield loss. The black or stem rust caused by Puccinia graminis triticale, and the brown or leaf rust caused by Puccinia triticina, and the yellow or stripe rust caused by Puccinia graminis triformis. Besides, the smut, loose smut, which is also very important, causing severe yield loss, which is caused by Ustilago triticale. The common bunt disease, which is caused by Tilletia caris or Tilletia foetida, and then kernel bunt or partial bunt, which is caused by Neovacia indica, and then powdery mildew, which is caused by Erysife graminis triticale. It is also known as Bloomeria graminis, and then take all disease caused by Ganoderma graminis triticale. Besides, the bacteria Ratheabacter, which is causing Tundu disease, which is found in association with the nematode Angina triticale. And let us see one by one in detail. The black or stem rust of wheat, which is caused by Puccinia graminis triticale, which produces a dark brown rusty pustules on the stem of wheat crop, which later turn into black color during teleospore formation. This is a heterogeneous type of rust, so which needs an another host to complete its life cycle. What is that another host? That is the barberry crop, and the recently developed severe strain that is mentioned as UG99, and people are trying to develop the wheat resistant variety by against this virulent strain UG99. So this disease which threatens the entire world, entire India, and causes severe yield loss. And this Puccinia graminis triticale, which is having 5 different stages of life cycle, and the stage 0, which is the spermagonial stage, and the stage 1, that is acial stage, both are on the alternate host, that is Barbaris vulgaris, while this stage 2, 3 and 4, which forms iridospore, teleospore and basidium, which are all found on the target host, that is wheat, which is a monocot in nature. And this is the typical life cycle of Puccinia graminis triticale. So the pathogen which the basidiospores which from the target host falls on the alternate host, where it produces pycnial as well as acial stage. Pycnial stage are formed on the upper surface of the leaf, which are flask-shaped structure, which is having a receptive hyphae at the tip of the flask structure, while the spermatia present within the flask-shaped structure that serves as the male unit, and the union of male and female unit which forms the dikaryotic mycelium. This dikaryotic mycelium which produces acium, which is corresponding to the pycnial stage on the under surface of the leaf. This acium or inverted cup-like structure which bears the dikaryotic hexagonal shaped aciospores. These aciospores carried through the wind and falls on the target host, that is wheat, where it produces uridial teleal stage, and upon the maturation of the teleal stage, the basidiospores formed, which again falls on the alternate host. By the way, the life cycle that repeats, while these iridospores are mentioned as repeating spores because within a single cropping period of the wheat, it can produce several generation of symptoms, and hence it is mentioned as repeating spores. This stem rust is favored by the hot days with the temperature of 25 to 30 degrees Celsius, and with the mild night temperature ranged between 15 to 20 degrees Celsius, and wet leaves from the rain or dew formation that also favors the development of pathogen. And then what is the mode of spread? The primary spread is mainly through windborne aciospores from the barberry and the iridospore from the wheat host, while the secondary spread is mainly bound by the iridospores from the wheat hosts. And then The disease is very severe and hence it has to be managed. Only thing is to develop the resistant varieties. There are some reported resistant varieties, that is DBW 17, PBW 550, LOK1 from Darwar, and then Karnal against in Darwar and Karnal. And then Barbary eradication is another important program by which we are eradicating the one of the life stages, two life stages from the wheat life cycle. While the disease is very severe, we can go for application of the combination fungicide, that is azoxystrobin plus cyproconazole at the rate of 0.05%, or else the tebuconazole, which is commercially called as tilt, and then tebuconazole plus triadimenol, and then tebuconazole alone or carboxin, which can be applied for the management of wheat stem rust. And then second important disease is wheat brown rust or leaf rust, which is caused by Puccinia triticina. Earlier it was recognized as named as Puccinia recondita form of Specialis tritici, so which affects all the wheat, barberry, and as well as the other species of barberries, and then which produces a small circular to oval brown rust pustules on the upper surface of the leaf. These pustules are scattered across the leaves randomly, and the spores develop on the leaves and leaf sheaths. What is the alternate host for this type of rust? That is Thalictrum species, which is commonly mentioned as common meadow rue. And then this is present in all the wheat growing areas within India. And then the pustules, which are formed brown in color earlier, will turn into black at the time of maturity. So these black sore are called as the teleospore. Microscopically, the teleospores of this Puccinia triticina is bicelled with rounded end. And then what are all the predisposing factors? The warm, humid weather that and with the intermittent rainfall favors the development of disease. Hot days with the temperature of 25 to 30 degrees and mild nights of 15 to 20 degrees, besides the leaf wetness from rain and dew, which are all favoring the development of disease. And here also the primary spread is mainly through asiospores from the alternate host and the iridospores, while the secondary spread is mainly through iridospores formed on the wheat host. And then yellow stripe rust, which is caused by Puccinia stripiformis. Why it is called yellow stripe rust? Because the rust pustules are yellow in color. They are arranged in a linear line on the leaf, and hence it is mentioned as yellow rust or stripe rust. So arrangement of these pustules are parallel to the veins on the leaf blade. And what are all the alternate hosts for this life cycle? That are Barbary and Mahonia. In India, the stripe rust is a severe threat, and in the larger area grown in larger wheat growing area in the northern part of India. Black pustules. So as I told you, the iridospores, which are yellow in color, while at the time of maturity these iridospore are converted into dark black pustules, which are all teleospore of the fungi, and these are all formed at the maturity stage of the pathogen, which is producing bicelled teleospore, pedicellate teleospores with flattened end. The next important disease is loose smut of wheat, which is caused by Ustilago tritici, which is mentioned as internally seed-borne disease. Why? Because it infects the embryo and remains in the embryo as a dormant mycelium, and all the grains in the airhead are completely converted into smut sore, and this smut sore that contains the spores, which are single-celled brown in color, mentioned as smut spores or teletospores. And here the life cycle that repeats. Those seeds which are infected with the Ustilago tritici and remaining as a dormant mycelium within the embryo that is remaining dormant as such in the embryo. When the seed germinates and the plant grows, the pathogen also germinates, grows along with the plant systemically, and expresses at the time of earhead emergence, which replaces all the spikelets into smut sore. And these smut sore contains numerous single-cell brown color smut spores, and these spores are carried through the wind and falls on the newly emerging flowers, and attached with the stigma enters through the style and reaches the ovary. There it remain dormant as a dormant mycelium, and when the seeds formed from this infected florets are used for sowing, then the pathogen also germinates along with the seed, and the life cycle that repeats. And the wind and moderate rainfall with the cool temperature of 16 to 22 degrees Celsius favors the development of this disease. And for the management, definitely we need to go for seed treatment, since it is internally seed borne pathogen. So the hot water treatment with 52 degrees Celsius of hot water and soaking the seeds in 11 minutes favors in the death of the organism present dormant within the embryo. Luthra and Satar, they have developed the solar treatment method, in which they mentioned soaking the seeds in the cold water for 8 to 12 hours and then drying the same seeds on the floor in the bright sunshine for 12 to up to from 12 to 4 p.m., which favors in reducing the disease incidence. Besides, using systemic fungicides like carboxin, benlate, etc., as a seed treatment fungicides, and then spraying of propiconazole 0.1% at the time of flowering, that are all helpful in reducing the disease incidence. A combination of Vitavax with the Thiram is very effective as a seed treatment fungicide. And then the next disease is hill bunt of wheat. This hill bunt or stinking smut or common bunt, which are caused by Tilletia caris and Tilletia foetida, and the entire kernel is converted into black sorus consisting of dark or brown moss teleospores. So these are all covered by a papery thin membrane called peridium, and this sorus is light to dark brown in color and called as bunt balls. And these are all the infected grains and the infected florets. And these bunted heads are very slender, bluish green, and may stay greener longer than the healthy airheads. And these bunt balls change into a dull grey brown at maturity, and at which they become very conspicuous in nature, and the fragile covering membrane of the bunt balls are ruptured as mentioned earlier as a peridium, which ruptured at the time of harvest and producing the dusty clouds of spores. These spores have a fleshy odor called caused by trimethylamine, a compound, specific compound called trimethylamine. And this is the life cycle of the hill bunt. So the life cycle of the hill bunt, the spores which falls on the florets, which invades into the floret and enters into the wheat earheads, and they germinates and establishes intracellularly and affect the individual florets and converted into bunt balls. These bunt balls, which are all released into the environment and produces the teleospores, which are all dikaryotic in nature. At the time of in the teleospores, the karyogamy followed by the meiosis that occurs. So during the after the karyogamy, the teleospores that germinate and produces basidiospores, and these basidiospores are unite together and form a H-shaped structure to form the dikaryotic infective hypha. This dikaryotic infective hypha, which affects the florets and remain in the florets as a infective and affect the florets, converted into wheat bunt balls. And this disease is externally seed borne, and systemic infection is also there, so which can be controlled by spraying of fungicides. Besides, as mentioned earlier in the loose mat, here also the seed treatment with the systemic fungicides like carboxin, Vitovax and benlate at the rate of 2 gram per kg, and spraying of propiconazole at 0.1%, which will be helpful in reducing the disease incidence, and use of resistant variety, which will be much more helpful, and the crop rotation with the suitable intervals, which is also effective in reducing the incidence at field level. And then powdery mildew, earlier it was mentioned as Erysipeia graminis DTC, and now it is renamed as Plumeria graminis DTC. So this pathogen known to produce grayish white powdery growth on the leaf blade, as well as on the leaf sheath and floral parts. Because of this infection, the leaf size and the number of leaves got reduced. So this fungi remain infected in the infected plant debris as a dormant mycelium, and in the dormant mycelium, which is having the chasmothecium and the ascospore from the chasmothecium, that forms the primary source of infection, and the airborne conidia that helps in the spread of secondary spread of this disease. And generally this powdery mildew are favored by warm humid weather. The temperature range between 20 to 21 degrees Celsius, and the humid weather favors development in a much faster rate. Application of wettable sulfur at 0.2% or even carbendazim at the rate of 500 gram per kg, which per hectare, which will be helpful in reducing the disease incidence. Kernel bunt or partial bunt, which is caused by Neovassia indica. It is first reported by Dr. Mitra from Karnal. And then these grains are converted either partially or wholly into a black powdery mass covered by the enclosed by the pericarp. And the teleospores, which are formed at the end cell of sporogenous hyphae, and the absence of sterile cells in the teleospore masses are the special characters of this Neovassia indica. And then these teleospores, which remain a long pedicel like appendages, and which germinate, produces a promycelium, basidium, that is basidium, which in turn produces promycelium, and this promycelium bears haploid basidiospores, maximum between 32 to 130 numbers. There is no H-shaped heterokaryon formation as we studied in the case of Tillechia. And besides, the absence of trimethylamine is also the another differentiating character from that of the wilt, the hill or common bunt. And this Neovassia indica produces two different types of sporidia: allantoid. Allantoid means banana shaped, and then filiform. Filiform means thread like teleospores. This is the long hyaline appendage got attached with the teleospores, and this teleospores, which germinate and produce promycelium, and at the tip of promycelium the basidiospores are formed, which range from 32 to 130 numbers. And then temperature of 15 to 25 degrees Celsius and relative humidity of 82% and presence of moisture that favors the development of this disease. And for the seed treatment, we can go for any of the seed treating fungicides which are effective against the smut or bunt, and then spraying of hexaconazole at 0.2% during the boot leaf stage, helpful in reducing the disease incidence. Treating the seeds with 1.5% sodium hypochlorite solution for 10 minutes will also be helpful in reducing the seed-borne infection or the initial infection. And removal of infected seeds are the best advised management strategy to avoid the incidents under the field conditions. And then next one is flax smut, which is caused by Urocystis agropyri. It is a smut fungi that affects the leaves, leaf sheath, culm and stem, and characterized by producing the chains of sori arranged in a linear manner on the plant tissues that later ruptures and releases black powdery mass of spores. And these streaks are very greasy in appearance, and this diseased plant still remain stunted and produce excessive number of tillers, and twisting of affected leaves and drooping down of the flag and flag leaf and withering of leaf are also the added symptoms to identify flag smut under the field conditions. And the fungi which causes the flag smut produces small spore balls which are consisting of 1 to 4 teleospores, and these spores are surrounded by the sterile cells, a layer of sterile cells. The teleospores are red to brown in color, smooth walled and round in shape, which germinate produces short hypha or promycelium, and this promycelium which in turn produces 3 to 4 basidiospores at the tip of promycelium. And for the management we need to go for seed treatment with the fungicides like carboxin or Vitavax. They are benlate at the rate of 2 gram per kg, and spraying of propiconazole at 0.1% will helpful in reducing the incidence. And the shallow planting is better than the deep planting to reduce the infection, probably because the seedling is in the susceptible stage for a shorter period of time only. 1 or 2 year break from cultivation of wheat may be beneficial, and then use resistant cultivars to avoid severe incidents. And then gloom blotch, which is caused by Septoria nodorum, it infects the glooms and develop gray brown blotches on the glooms. This Septoria leaf and gloom blotch pathogen, which survives within the infected straw or seed and volunteer wheat crops, serves as a source of inoculum with a new crop for the new crop to develop the infection. And this infection occurs during the periods of extended wetness, and it is a pycnidial fungi, produces pycnidiospores which act as the inoculum. The windborne conidia and splashes of rain are helpful in spreading of this disease. And better go with the seed treatment, go for the seed treatment with the carbendazim at the rate of 2 gram per kg, as well as foliar application of propiconazole. The commercial name is tilt at the rate of 0.1%, which is helpful in reducing the incidence. The next important disease is take all disease, which is caused by Gammonomyces graminis formospecialis triticea. So, which is initially causing the rotting of roots as well as the basal portion or lower portion of the stem. This basal portion of the stem and leaf sheath tissues and roots, they may turn into shiny black in color due to the infection. The affected plants are stunted in the growth, and they with the whitened stem as well as the spikes. So for management we should growing legumes uh, instead of grasses in the surrounding area, which will be helpful. And then seed treatment with the biocontrol agents like Pseudomonas or Bacillus subtilis, which will be helpful in reducing the initial incidence. Among the bacterial diseases, the bacterial streak caused by Xanthomonas translucens is very important, and which produces water soaked narrow chlorotic spots or lesions or stripes on the leaves which are parallel to the veins. In severe cases, it produces yellow color sticky exudation due to severe infection of the bacteria, and this exudates which dries to form a crusty droplets or translucent film on the surface of the leaf area. Besides, it also produces chaffy symptom on the wheat heads due to the infections. For the management, you should go for the crop rotation with the non-host crops, or else treating the seeds with the acidified cupric acetate at the rate of 0.5%, and or treating the seeds at 45 degrees Celsius for 20 minutes will be helpful in reducing the disease incidence. And then next important disease is the gloom blot, head blight or scab, which is caused by Fusarium graminearum. Generally, the Fusarium are affecting only the root crop as well as infecting the vascular region of the plant. But this Fusarium graminearum, which mainly infects the inflorescence and causes damage to the grains. premature bleaching of one or more spikelets in the entire immature wheat earhead. This bleaching can start anywhere on the earhead and spread until the entire earhead is bleached. And these bleached spikelets are sterile or contain shriveled, discolored seeds. During humid conditions, white or pink color fungal growth can or with orange spore mass can be seen on the bleached spikelets. Blue or black colored perithecia will also be formed on the airhead. This Fusarium graminearum also produces a toxin called deoxynivalenol, or it is mentioned as a devoin or vomitoxin, which is a serious mycotoxin and a threat to the human being. This is the typical bleaching symptom formed on the spikelets of wheat earhead due to Fusarium graminearum. And when during the severe period, it also produces white mycelial growth and pinkish or orange spores production on the infected earheads. Take all disease which is showing white earhead, while this scabby growth as well as presence of mycelia and then bleached earhead, which will be differentiating from that of the earhead or white chaffy earhead produced by the wheat take all disease. And then leaf blight disease, which is caused by Alternaria triticina, though it is a minor disease, it is to be given importance because every minor disease will become a major disease at the later stage. So this pathogen known to produce reddish brown oval spots on the younger seedlings with a bright yellow margin, and this pathogen having the potential to become a externally seed-borne as well as internally seed-borne pathogen. And the temperature 25 degrees Celsius, which will be helpful, and high humid weather, which will be helpful in development of the disease. Treating the seeds with hot water at 52 degrees Celsius for 10 minutes will helpful in controlling the pathogen. Or the use of resistant varieties like Arnatka or E6160 or K7340, which will be helpful. If it is observed at the later stage, then apply Mangozeb at the rate of 1.5 kg per hectare to reduce the incidence. And this is the typical spores produced by the Alternaria, and it appears in the field when the crop is at 7 to 8 weeks old. And we have already studied the development of symptom, that is formation of brown oval spot on the leaf, and the severe and several such spots coalesce together cause complete drying of leaves, which will give you the burnt up like appearance. And then next leaf blight, which is caused by Helminthosporium species or Helminthosporium leaf blotch, and this Helminthosporium are bipolaris, which is common on the seed, stem, all the parts of the plant, while the Drusillera, which is the another form of Helminthosporium, which is common on the leaves, stem, and less on the seeds. This pathogen is known to produce small brown spot initially. Several such spots coalesce together and cause complete blighting of leaves. So during the tillering stage, it initiates from the lowermost leaves and then spread to the entire plant, leading to complete blighting of all the leaves. Finally, the infection that covers all the plant. And for the management, you can use enough organic manures and cultivate wheat in the fertile land with the healthy seeds. That are all the primary requirement. And then seed treatment with the carbendazim plus thiram combination at the rate of 2 gram or tebuconazole 2% at the rate of tebuconazole at the rate of 1 gram per kg of seed, or Vitovax 200 at the rate of 2 grams per kg of seed are advised to reduce the seed-borne infection. Use balanced fertilizers and planting at optimum date, and use some of the reported resistance varieties like Triveni, Achua, Rohini, or Braikuti, etc. When the disease appears at the later stage, you can go for application or spraying of propiconazole at the rate of 1 ml per liter, and repeat based on the necessity at 15 days interval. And then tan spot of cereals, which are also a emerging disease, and this produces a typical tan brown lesions surrounded by yellow halo. The mature tan brown lesions have a dark area at the center, and these lesions merge together and causes complete blighting of the entire leaves. The fungi that causes tan spot that survives in the infected plant debris of the previous crops. They produce small reproductive structures on the infected plant debris during spring season. And the pathogen which is causing the tan spot are Pyronophora tritici rependis. The synonym is Helminthosporium tritici rependis, which produces two types of spores. The asexual spores of conidia are produced mainly during and the maturing lesions of on the developed on the leaves, and these conidia are hyaline to light brown, cylindrical, and have 9 transverse septations. The conical tapering of the basal cell of the conidium, which gives appearance of snake head, which is the diagnostics of Drusillera tritici rependis. And the sexual spores are ascospores, which are produced within the ascocarp pseudothecia, and these ascospores serves as the primary source of inoculum. And the next important disease is Thundu disease or bacterial spike blight, which is caused by Rathibacter tritici in association with the nematode called Anguina tritici, which causes yellow ear rot. So this Rathibacter tritici early in the field it produces yellow ear rot or Thundu disease, which are producing yellow or white streaks along parallel to the veins. Later, a conspicuous yellow bacterial exudate that appears on the wheat head. This is the diagnostic symptom, specific diagnostic symptom for this disease. The heads and necks frequently become distorted with the sticky bacterial masses. At an earlier stage, the leaf may wrinkle or twisted, and they emerge from the bacterial world, and when dry the exudates appear as a white flux on the affected heads as well as on the leaf surface. The affected seedlings are very stunted in nature. So as I told you, this Rathibacter is also associated with the seed gall nematode Anguina tritici, which is a due to this infection, the infected plants are stunted in nature. Their airheads and leaves are linkled, twisted, and the entire kernel is replaced with the galls filled with the Anguina tritici nematode. This is the typical symptom of galls formed on the airhead, and these galls are infected, and these are all filled with the nematode Anguina tritici. And what is the management strategy? The hot water treatment of this contaminated wheat at 54 to 56 degrees Celsius for 10 to 15 minutes will be helpful. Pre-soaking the seeds before the hot water treatment has also been advocated to get the better effect of hot water treatment. And the seed can be soaked in the water for 2 hours and then heated up to 51 degrees Celsius for 30 minutes, helpful in reducing the incidence. And then the cylinder machines are physically the affected gall infected nematode infected galls, seed galls have to be removed, separated. So you have to use disease free seeds or plant, and then planting in a disease free soil, clean seed, and the complete disease pathogen free soil which favors reduction in the disease. Soil treatment with the carbofuran is also recommended at stages. Thank you.